Right. Hello, everybody. This is a great pleasure to introduce today's speaker, Dave Krause. He studies evolution of the competition for the scarce resources using the rooting yeast as a model. He got the PhD in University of Illinois Urbana-Champaign in the Professor Rachel Whitaker's lab, and he joined the Chris Hittinger's lab since 2016. Since then, he's been very active and contributing to the field so much. He actually selected for the speaker for the genetic colloquium actually 2 years ago, and now he's back and giving his most recent, sharing the most recent research with us. So I'm very excited to hear his story today. And welcome, Dave. Thank you so much.
Thank you, Aki. All right, so I'm going to let this load up for a second. Here we go. All right, so I'm going to talk today about the evolution of anaerobic growth in budding yeasts. Thank you, Aki, for that introduction. I'm really excited to be here and share some of this latest work that I've been doing in Chris Hittinger's lab over in the Wisconsin Energy Institute building. But sort of to start as a background, I'm generally interested in how organisms, but especially microorganisms, compete for scarce resources. So resources are generally not in massive abundance, and they're constantly fought over with fierce competition. And so the first project that I actually did in Chris's lab was study the production and utilization of a siderophore, which is used to scavenge for scarce iron in the environment, and its production by a variety of yeast species, where we figured out the genes that were involved in its production and actually looked into some cool evolutionary stories along the way of how these genes have evolved. And of course, most of all life needs iron to survive, so the mechanism that microbes and other organisms often evolve is to sort of compete for it and scavenge for it. On the other side of this slide is what I'm going to talk about today, which is a little bit of a different story. It's growth in the absence of molecular oxygen. And as I'll talk about, molecular oxygen is not actually, O2 is not actually required for growth in many organisms. And therefore, it doesn't necessarily need to be scavenged and competed for. In fact, in many cases, it can just be, with the right evolution, ignored altogether. And so, anaerobic growth is found in all domains of life. There are several bacterial lineages and archaeal lineages, I should say more than several, just countless lineages of these organisms that are capable of growing in the complete absence of molecular oxygen. And although we often think about multicellular eukaryotic organisms as generally like us requiring oxygen, there are actually lots of examples of especially unicellular eukaryotes that are capable of growing without oxygen. And this makes quite a bit of sense in the case of bacteria and archaea, whose evolution likely spent a lot of time before the great oxygenating event, before oxygenic photosynthesis arose. And so there are countless examples of metabolisms and biosynthetic pathways that just have never experienced oxygen. Meanwhile, the origin of eukaryotes likely comes sometime after that oxygenating event. And so although I'm not going to talk much about any sort of controversies or interesting research about the origin of eukaryotes, suffice to say that it's often generally thought that there are a lot of obligate aerobic eukaryotes and a lot of anaerobic eukaryotes that have sort of evolved ways to grow anaerobically after the fact. And so budding yeasts kind of fit this profile. They're generally obligate aerobes. They need oxygen to grow for some purposes, whether that is their general metabolism or biosynthetic pathways, which I'll get into later. There's only two lineages that we really know of that are capable of growing under anaerobic conditions, and I should say growing and propagating stably in an anaerobic chamber is the way that we've tested this. And so this is a large tree of over 300 species of budding yeasts built on their genomes. The Hittinger lab has built this really nice tree that shows the relationships of over 300 species of budding yeasts. At the sort of bottom left of that tree, I've pointed out Saccharomyces cerevisiae, the model yeast that we all certainly know. And it's only a group of species that are closely related to the Saccharomyces that are actually capable of growing under strict anaerobic conditions. And you have to go all the way out to this fairly divergent lineage at the top right of that tree, the Britannomyces, which many people have probably heard of one way or another. That's the only other lineage that we really know of that can grow under strict anaerobic conditions. So this is something that's likely evolved or emerged twice independently in the tree. But for the most part, most of that tree space is obligate aerobes that require oxygen. Yeah. Oh, that's not great. Let me see what I can do. Duplicate slideshow. Maybe that's my problem. That should fix it. Okay. Yep. Sorry about that, guys. Okay. You did miss my pretty picture of a plate of yeast. And so, but most of what I've said, I've done without visual aids apparently. So I will skip back to where I was. We haven't missed too much. So like I said, budding yeasts are generally obligate aerobes, but there are a couple of lineages of yeast that appear to have evolved the ability to grow anaerobically. And so I'm fairly interested in this topic, not only because if we really were to better understand why and how these yeasts have evolved the ability to grow anaerobically, we might better understand yeast ecology in general, because oxygen is something that you're either interacting with or not interacting with in the environment. But also, what if we could engineer some of these aerobic yeasts into anaerobes? I think there would be a lot of industrial implications and ways of applying that in the industry, because there are a lot of interesting yeast species out there that we know to be obligate aerobes that actually do a lot of interesting metabolisms that would be very useful, whether that is oleaginous yeast that make lipids, whether it's vigorous xylose fermenters. So a lot of yeast don't naturally ferment xylose. There's actually only relatively few that are capable of doing this, but they don't grow anaerobically. And also some work that I've done before on these yeast species that produce pulcaramine that may have interesting branched chain amino acid flux that we could implement into biofuels. Those species don't grow anaerobically either. And so a lot of the time when we're looking at industry, while there are people that are using a lot of different yeast species to do very cool things, a lot of the time we're engineering Saccharomyces cerevisiae to do have some of these traits that we find elsewhere in the tree of budding yeasts. So anaerobic growth presents certain physiological challenges to yeasts. So there's a reason that it has evolved twice, but for the most part, the canonical yeast pathways have issues, whether that's the production of uracil, NAD, the production of ergosterol, an important sterol in yeast cell membranes, or unsaturated fatty acids like oleic acid, which is required for cell membrane structure as well. It turns out that all of these require oxygen at some level to produce these molecules. And of course, there's the one that we think about a lot when it comes to energy generation in the cell, respiration. And so for yeasts, as we understand it, we only know of using oxygen as a terminal electron acceptor for respiration, although there exist countless examples across the domains of life of using some alternative terminal electron acceptor. We only know of oxygen for respiration in yeast so far. And so you might think that Saccharomyces cerevisiae has evolved some way of getting around these problems, and that's why it can grow anaerobically. And in fact, that is mostly the case. So Saccharomyces cerevisiae has the ability to do a totally balanced ethanol fermentation. It doesn't need respiration to grow. And in fact, when you grow Saccharomyces cerevisiae in the lab on glucose, they repress respiration and solely generate energy via substrate level phosphorylation through fermentation. They can make uracil anaerobically because they actually are part of a group of yeasts that obtain via horizontal gene transfer a bacterial gene that replaces the step that normally requires oxygen and instead uses a different terminal electron, sorry, not terminal, but a different electron acceptor for that reaction. Further, they can salvage niacin from the environment and actually put that into a salvage pathway that enables them to make NAD, whereas the normal biosynthetic pathway requires molecular oxygen at several steps. They're actually able to sort of salvage this from just exogenous niacin that we can put in the medium or they might find in the environment. If we give them unsaturated fatty acids, they're able to actually just diffuse those up through their membranes. And so they no longer need to produce them when they're supplied with for example, oleic acid, which we'll often add to the medium when we're growing them anaerobically. And finally, for sterols, they actually have this transporter-mediated uptake of exogenous sterols. So if we put sterols in the medium and grow Saccharomyces cerevisiae anaerobically, they will uptake these through dedicated transporters. So they sort of have a way around all of these physiological things as we understand them to be important for anaerobic growth. But I should say before I go forward too much further that this list probably isn't exhaustive. This is just sort of a rough list of things that people have really thought about in the past several decades of studying yeast and anaerobic growth. But there have been things like gene deletion libraries that have been screened under anaerobic conditions. People have done gene expression analyses under anaerobic conditions. And ultimately, these suggest that there could be other pathways out there, as well as quite a bit of regulation that is involved in anaerobic growth. You might imagine that organisms that grow anaerobically, like Saccharomyces cerevisiae, have a totally different transcriptional profile that they undergo under anaerobic conditions, whereas we don't really understand how a lot of these obligate aerobic organisms respond to an anaerobic stress or environment. So I talked about all these different ways that Saccharomyces cerevisiae can grow under anaerobic conditions. And one of the first things that I wanted to do was compare the genes that underlie those particular processes against sort of all of the different species that we know can grow anaerobically versus can't grow anaerobically to try to determine which genes were really responsible or are the most responsible for this trait. And so I had done this before with some of my work in the yeast siderophore pulcherrimon. And the idea is, or was, that we could didn't realize that was showing up. So the idea was that we could take all of the yeast species that we had sequenced at the time, which was about 100 that were publicly available, look for pulcherrimon producing species in the lab, which are labeled here in the top row as a red square. And then ultimately we looked for genes using blast similarity of other genes that have been known to be involved in pulcherrimon production in bacteria, which unfortunately there were not really good homologs for that. But ultimately, we were able to find a gene cluster that correlated fairly well with the production of pulcherrimon. And by doing some genetics, we confirmed that these two genes, PUL1 and PUL2, were involved in the biosynthesis of pulcherrimon. And when we looked back at the tree and compared who could and could not make pulcherrimon with who did and did not have PUL1 and PUL2, there was a pretty good correlation. There are some species that have both of these genes but don't make pulcherrimon, but we never found a species that could make pulcherrimon but lack these biosynthetic genes, which suggested that you need these genes to make pulcherrimon. And so we wanted to apply sort of a similar approach to the genes that underlie some of these pieces of anaerobic growth. And so when we look at sort of a larger tree, this is a bit of a larger tree, but it's on a smaller set of species. So these are all the Saccharomycetaceae, which is a set of yeast species that are closely related to Saccharomyces. And it's just a cladogram view of them. I've put in gray as one of those columns whether or not the species can grow under anaerobic conditions as we've tested in the lab. And that growth is limited to 4 genera: the Saccharomyces, the Nicasiamyces, the Kazakhstania, and the Naumovizyma. But it's 3 genera closely related to the Saccharomyces genus. So I talked about that Euro-1 gene that had been transferred into Saccharomyces and a set of relatives that enables them to make uracil under anaerobic conditions. Well, as it turns out, virtually all of the Saccharomycetaceae have this Euro-1 gene. And so it doesn't seem, at least initially, that Euro-1 is a major factor in anaerobic growth, considering there's a lot of species that don't grow anaerobically that have this gene. And also there's a set of Nicasiamyces species that are missing Euro-1, so they actually don't have this uracil biosynthesis gene. In principle, they could uptake uracil from the environment. They don't necessarily have to make uracil under anaerobic conditions, but it's unlikely that the ability to make uracil under anaerobic conditions is a driving factor behind anaerobic growth, since so many species have this gene but do not grow anaerobically. And the same is true of these NAD salvage genes. It actually turns out that most budding yeasts in general have the genes that are involved in salvaging NAD by using niacin from the environment. So it's really hard to say that this is a good explanation for the rise of anaerobic growth, because it's generally found in all budding yeast species that we've sequenced. The final thing that I mentioned that has a genotypic, something underlying it that's genotypic, was sterol transport. We know of 2 sterol transporters in Saccharomyces encoded by the genes AUS1 and PDR11. And when you look at which species have this versus which species don't, it looks like a better correlation than any of these other genes, in that all of the anaerobes have AUS1 or PDR11, and several species that don't grow anaerobically have them, but it's definitely limited to a series of yeasts that are fairly related to Saccharomyces. So of all the physiological understandings that we have of Saccharomyces and its anaerobic growth, this seemed like the best fit to try to study it a little further. And something kind of interesting that I found sort of during the early pandemic quarantine time that I hadn't spent very much time studying prior to that. So in a way, a very small side benefit to our time quarantining and doing a lot of computational work, was doing a bit of research on this idea that the sterol transporters alone are actually not sufficient for sterol uptake. So as it turns out, a group tried to constitutively express one of these sterol transporters, Saccharomyces cerevisiae, and then expose the yeast to a drug that stops their ergosterol biosynthesis, ultimately requiring them to uptake sterols to grow. And if you constitutively express the sterol transporter, you actually can't get sterol transport to occur, and you can't get the strains to grow in the presence of this drug. And as it turned out, they had some inklings about what they might want to test out alongside just constitutively expressing this sterol transporter. And so they expressed this cell wall nanoprotein DAN1 alongside this sterol transporter, and it helped a little bit. I will say that the title is a little misleading to say that it's totally sufficient for the process. The co-expression of this nanoprotein increased sterol transport, but it didn't actually increase it to the sort of biologically relevant levels you would expect to actually overcome the drug inhibition. So it seemed like the cell wall nanoprotein was helping, and I think moving forward, we can sort of believe that, but that is just one single nanoprotein, and I'll get more into that later. And then there was also a study in Candida glabrata, which is fairly closely related to Saccharomyces. It fits in that Nicasiamyces sister genus to Saccharomyces. And they found that if you delete a single one of these specific cell wall nanoproteins, you can actually abolish sterol uptake under several conditions in Candida glabrata where they normally uptake sterols. So it seems like, looking both ways, expressing one of these cell wall nanoproteins can help with sterol transport, and deleting one of them in a different organism can actually get rid of sterol uptake. So it seemed like these cell wall nanoproteins are doing something in sterol uptake. We also know that they're anaerobically induced in Saccharomyces cerevisiae. So there's this study that looked using northern blots, studying the expression of some of these cell wall nanoproteins, found that actually under anaerobic conditions, there's a set of, I believe they're showing here 8 genes that are not really expressed under aerobic conditions on the far left there, the O2, and then when they're transferred into nitrogen gas, which allows them to go anaerobic, you see that you start to get this induction of all of these cell wall manoprotein genes, at least their RNA. But at the bottom are actually the cell wall manoproteins that are normally expressed under aerobic conditions in Saccharomyces cerevisiae. And while they start out fairly strong when they're exposed to oxygen, as the cultures go anaerobic, we start to see those fade away. So we almost see this replacement of the entire cell wall manoprotein set of genes, at least at the transcriptional level, as the cultures go anaerobic, you transfer from these aerobic proteins to these anaerobic proteins. And we've also seen similar stuff, just looking through some RNA-seq data so that it's not just this one experiment. We do see a similar pattern using RNA-seq as well. This data taken from a recent paper from UW-Madison and collaborations with the GASH lab. And so sterile transporters, to kind of finish this topic of the cell wall manoproteins and why we think they're interesting, not only do they have, is there some genetics behind it, and not only is there some gene expression happening that seems to be involved with sterile transport, they're also physically clustered in the genome with cell wall manoproteins, these sterile transporters are. And so if you just look at a few species here that I'm showing at the left, just sort of rough diagrams of what their genomic neighborhood looks like for these cell wall manoproteins, they're often next to PDR11 or AUS1, these genes I described earlier as being sterile transporters. And so there is just this physical gene clustering that just seems interesting and is very suggestive that for some reason these things are being localized together in the genome. What that reason is, I think is interesting, but it needs to be further studied. And as a quick example, this is what one of those cell wall manoproteins looks like at the amino acid sequence on the right. This will become important a little later when I talk about some of the computational stuff that I've done. But ultimately, they're characterized by this N-terminal signal sequence labeled in gray, a couple of fairly conserved motifs labeled in yellow. But then what makes them fairly difficult to align together, at least at the whole protein level, is that in green, roughly half the protein is just this really rich region of serines and threonines. It's very low complexity. And part of the reason for that is that these serines and threonines are typically O-manosylated, which is a process that is undergone by serines and threonines in these proteins. And so they're covered in mannose residues. And finally, this C-terminal portion is fairly conserved. It's a very hydrophobic portion that's just part of getting it to the cell wall. So I had mentioned the studies that have been using genetic evidence to figure out a role for the cell wall manoproteins. And there are two others that I didn't mention, but they were done directly in Saccharomyces cerevisiae, and there was a bit of conflict. So this is what sort of primed me to try to study these for myself. Two different studies. The first study just basically said in the text that you could delete these three different cell wall manoprotein genes, tier 1, tier 2, and TIP1, and you have no detectable phenotype when grown anaerobically. And then on the flip side, another lab had deleted tier 1, tier 3, and tier 4 individually, not in the same strain, just individual deletion mutants, and found that all three of those deletion mutants were unable to grow anaerobically. So it sort of raised this question, are these genes really redundant and you can knock out several of them and still get anaerobic growth? Or are they generally very important? If you knock out a single one of them, you're going to lose anaerobic growth. And so I decided to undertake this experiment essentially earlier this year to go in and try to solve this problem for myself, delete these genes, all using various drug markers or selectable markers to construct a full 7-gene deletion mutant. So I should just mention actually real quick, Saccharomyces cerevisiae has 7 of these cell wall manoprotein genes in its genome. They're found on 5 different chromosomes. And we went ahead and went in and knocked them out. Genes that are colored the same color were deleted in the same, deleted in tandem at the same time because they were right next to each other. And so to sort of facilitate the usage of some of these markers and make this a little easier, we did delete DAN1 and DAN4 and tier 2 and tier 4 sort of as 2 genes at the same time. But ultimately, we constructed this 7-gene deletion mutant. And then we wanted to test all possible combinatorial mutants. And so what we did is we back-crossed it against a wild-type strain, sporulated that out, and collected all the possible combinatorial mutants. In the case of the 5 different loci that we deleted, although we deleted 7 genes, we deleted them in 5 separate loci. We wound up with 32 different strains that we then went into the anaerobic chamber and tested for anaerobic growth. And so here is just sort of a nice colorful figure demonstrating what happens when we test these strains in the anaerobic chamber. Every column is a different strain. And if a box is colored, that strain contains this particular gene. And if the box is grayed out, that means that that gene has been deleted in that particular strain. And so we can look at all the combinations, and at the bottom in purple is whether or not they grow under anaerobic conditions. And so a dark purple box means that they grow normally. And this is actually, it turns out that this experiment does not have a lot of intermediate phenotypes. The strains either grow just like wild type grows, or they don't grow at all, with the exception of those 2 sort of lighter purple boxes, which is 2 strains that grow fairly poorly, but are able to grow. They just reach a lower OD and they grow a bit more slowly than the other strains. There really were only these 2 intermediate strains. So first, tier 3, we find that tier 3 is actually required for anaerobic growth. If you delete it and it's the only deletion you make, then you abolish anaerobic growth right off the bat, and all tier 3 mutants fail to grow in the anaerobic chamber. Then there's this other gene, tier 1, that seems to be required for sort of a normal growth rate. If you just delete tier 1, or you delete it in a particular background, like the 1 deletion background, you actually are capable of growing anaerobically, but the growth appears to be quite poor. And if you continue to delete genes from that genotype, they no longer grow anymore. And what's kind of shocking is that these 2 genes are the most important from a deletion perspective. If you delete them, you get the strongest phenotypes. And yet, they also are together sufficient for anaerobic growth if these are the only 2 genes you have in the genome. So this sort of turned on our head the initial hypothesis we had going into this, where we thought that there might be quite a bit of redundancy among these genes, and that while you might be able to delete 5 of them and still get anaerobic growth, we were thinking going in that you might be able to just delete any 5 of them and still get anaerobic growth. But as it turns out, there are these 2 genes, tier 1 and tier 3, that actually contribute quite a bit to anaerobic growth. They're probably the most important genes as far as the cell wall nanoproteins are concerned in this process. And so we wanted to test whether tier 1 and tier 3 were actually contributing different functions to anaerobic growth in some way, or whether if we were to overexpress one of these genes, or really any of these genes, we could sort of rescue anaerobic growth in these deletion mutants. And so this was sort of another test of this sort of redundancy model, the idea being that maybe these loci, if they were overexpressed, could yield anaerobic growth, and that you might only need a single or 2 cell wall nanoproteins. It's just that they aren't expressed high enough in some of these mutants. So what we did is we took all of these cell wall nanoprotein genes individually with their native promoters and terminators, and we cloned them into a high copy vector, and then put them into that cell wall nanoprotein deletion background that has all 7 deleted. And as it turns out, none of those strains grew in the anaerobic chamber. So overexpressing a single cell wall nanoprotein does not give you anaerobic growth. So we reasoned that maybe you need tier 3 for some reason. It was this essential gene that if you delete it, you don't get anaerobic growth. So maybe if we leave tier 3 in the genome and then try this experiment over again, we might see this coming out and working for some of these overexpression strains. And so we did that. We put these plasmids into the tier 3 competent background and didn't get results that were that surprising. So the overexpression of tier 1 enabled growth, but as I showed you before, having tier 3 and tier 1, we already knew conferred growth to these yeasts. But what was kind of interesting was that if you overexpress tier 2, this other gene that we've been normally deleting without any real consequence for the cell, when it's overexpressed in a tier 3 competent background, that strain could grow. So conclusion so far, based on this genetics work, is that tier 3 is required for anaerobic growth. Tier 1 seems to be required for some sort of normal anaerobic growth rate, but it's not explicitly required if some of those other cell wall manoproteins are present. Tier 3 and Tier 1 make up this minimal gene repertoire that you can use for anaerobic growth in Saccharomyces cerevisiae, it looks like. However, Tier 3 and Tier 1 are not interchangeable just by overexpressing one or the other, which seems to mean that they likely have some sort of divergent functional roles and are doing something else, are doing different roles in the cell. And actually, if you have Tier 2 being overexpressed with Tier 3, you get growth, implying that Tier 2 may be some sort of cell wall manoprotein that is functioning maybe like Tier 1, but ultimately its sort of native expression in the cell is sort of the reason that we're not seeing it have very strong effects during the genetics experiments. So now I'm going to move a little bit into the evolutionary side of this, with the big question being, when we first started all of this, why do some species have so many cell wall manoprotein genes? And the reason I say this is there are lots of species that have a single cell wall manoprotein. And it's often in the Saccharomycetaceae, it's in a single locus. Here I'm showing the gene neighborhoods of some of these cell wall manoproteins that we identified. And while several species just have a single one, and if you look at cell wall manoproteins out throughout the entire tree of budding yeasts, you often find that they have one or two of these cell wall manoproteins. Saccharomyces cerevisiae, like I said, has 7 of these, and they're in totally different genomic locations. Although several of these manoproteins look like they have genes nearby that look a bit like these other species, some of them are in totally different locations altogether. And so I'm going to talk briefly about this, this whole genome duplication event that occurred in budding yeasts that actually gave rise to the lineage that includes Saccharomyces cerevisiae before I go any further with some of this evolutionary stuff. But there's been some really cool work sort of in the recent past about how this whole genome duplication event occurred, and using tracking the evolutionary history of some of these duplicates in the Saccharomyces cerevisiae genome. This group was able to sort of support this hypothesis that rather than just writ large genome duplication event that occurring along the line of the Saccharomyces cerevisiae lineage, it likely actually occurred by a hybridization event between 2 parents. One that was an ancestor of the, as I'm showing on this tree, an ancestor of the Clavaromyces and Lachantia genera. And that ancestor likely hybridized with an ancestor of the Zygotorilospora terulospora genera. And this hybridization event ultimately gave rise to the lineage of yeast containing Saccharomyces cerevisiae, as well as the Acasiamyces and the Kazakhstania and the Naumavazyma and these Tetrapicispora that I'm showing on this tree. And so they, and at least initially, contain lots of gene duplicates, although from 2 different sources that over time have been either lost or in many cases maintained after a series of genome rearrangements. And so this is sort of the explanation for how this whole genome duplication event happened. And I'm going to refer to this a few times moving forward. So what we find is that this whole genome duplication event coincides with an amplification of these cell wall manoproteins. So I had mentioned this is that same tree as I've been showing, and I've just kind of bolded the lines of those species that underwent the whole genome duplication or are descended from that one that underwent the whole genome duplication. I have anaerobic growth shown in gray here. It's a subset of those that underwent the whole genome duplication. And then I'm showing again the distribution of those AUS1 PDR11 sterol transporters. And then to the right is just sort of a bar chart of the count of cell wall manoprotein genes in each of these genomes. And in these non-whole genome duplication lineages, generally they only have 1, although there is a case where there are maybe 2 or 4 of these genes. They're all kind of in the same locus in that case. But if you look at the post-whole genome duplication lineage, they have typically a lot. There can be as few as 3, but there can be as many as, I believe, 11 or 12 cell wall nanoproteins in a particular genome. So there was some kind of amplification that really happened after the whole genome duplication. And so because of that, we wanted to construct a phylogeny of these cell wall nanoproteins. I know we've been finding them using BLAST, but we wanted to assess their relatedness to one another. And we also wanted to see whether we could find specific alleles coming out as being more related to one another, and whether that corresponded with the gene synteny of these particular alleles in their genomes. And so this is a maximum likelihood tree using nucleotide sequences of these cell wall nanoprotein genes. And the branches are colored in many cases. And this coloration is based on their synteny with the Saccharomyces cerevisiae genes. So basically, if a gene is found in Saccharomyces cerevisiae, surrounded by particular genes, and we find those same genes in the vicinity of a cell wall nanoprotein gene from another organism, we've colored them the same color. And ultimately, we find that there actually are what look like sensible clades that represent particular homologs of these Saccharomyces cerevisiae genes. And what's also fairly interesting is that in the case of tier 3, for example, we see that at the root of tier 3, although this is fairly weak bootstrap support, we see that the Lachantia alleles, which are these non-whole genome duplication species, are found, they're clustered in with the tier 3 genes. And actually in tier 1 and tier 2 and tier 4 in blue and green there in the middle of the tree, we find that at the base of that clade is the Zygotorulospora and Torulospora gene sequences. But we also see there's like this entirely divergent lineage in the DAN called the DAN genes that we don't know much about exactly what they're doing, but they do appear to have diverged significantly since the whole genome duplication from the rest of these genes. And so far what we know about just from the phylogeny and synteny, though, what we're kind of excited about is that it looks like tier 1, which is that gene that I've already mentioned was fairly important to anaerobic growth in Saccharomyces cerevisiae. It's part of the same clade as tier 2 and tier 4, which is interesting because we saw that an overexpression of tier 2 sort of looked like it could serve the same function as tier 1. They likely originate from that ZT ancestor of the whole genome duplication event. And it's possible, although we still need to do more testing of this phylogeny, that tier 3 may have originated from that KLE ancestor of the whole genome duplication event. But what's really important from this tree as well is that these duplications of these cell wall nanoproteins seem to be ongoing. They're frequently happening within lineages, and so they seem to be just frequently duplicating. And these DAN genes, which had been considered very important, I talked about that study earlier, where they were looking at the DAN1 gene in Saccharomyces cerevisiae. It doesn't seem to contribute very much to anaerobic growth in the lab, and it appears to be really divergent from these other genes. And from what we can tell, it's also undergone just massive translocation and is usually only found in like subtelomeric regions of chromosomes now. And then TIP1 is also seems to be the result of some kind of translocation event where it wound up somewhere totally different from the rest of these cell wall nanoproteins, but we're still pretty unclear about what TIP1 is doing as well. And so to kind of summarize and bring together the evolutionary analysis with the genetics analysis that we've done in the lab, we think it's fairly interesting that the genes with the best correlation to anaerobic growth are also the most important in the lab. So we found that tier 3 and tier 1 were the most important genes for anaerobic growth in Saccharomyces cerevisiae. And if we look at the species that can grow anaerobically, all of them have a tier 3 homolog. And all but one of them have a homolog of the tier 1, tier 2, tier 4 complex. We're unclear what that one, what is going on in that one particular species, that's some Kazakhstania species, but we'll be looking further into that going forward. But for the most part, we can see that these DAN genes are actually really highly amplified in this Tetrapicispora lineage over at the far left of the whole genome duplication clade. They don't grow anaerobically. They don't have any tier 3 or tier 1 homologs from what we can tell, but they do have a lot of cell wall nanoproteins. We don't really know what those cell wall nanoproteins are necessarily doing, but they seem to have this diverged set of nanoproteins. So now I'm just going to finish up by talking briefly about sort of what we've found so far and how we're trying to determine whether they're consistent with various models of gene duplication. Since budding yeast kind of give us this nice tool model system to test these models. We initially started with this positive dosage model, as I had mentioned before, where perhaps in the ancestor, there was this whole genome duplication event, and then these cell wall nanoproteins became massively amplified somehow. And ultimately, this sort of increased dosage and increased expression of these cell wall nanoproteins somehow contributed to anaerobic growth. But we've sort of come to reject this model for the most part because it looks like individual cell wall nanoproteins seem to evolve their own functions. And you can't just delete any particular combination of these and be left with just a few. You have to be left with a very specific few cell wall nanoproteins to still get anaerobic growth. However, this other model that I'm very interested in testing in the lab right now, and we're sort of working towards it, is this idea that perhaps the ancestral cell wall nanoprotein locus diverged over time, and as it went into these two different lineages, the KLE lineage and the ZT lineage, the two ancestors of that whole genome duplication event, perhaps they had been functionally diverging to some extent, and ultimately when the whole genome duplication hybridization happened, you then got a strain that had these two really important genes for anaerobic growth, and potentially these are what really help to confer this particular aspect of anaerobic growth to that particular species or strain. And so I'm interested in testing whether some of these alleles that we can get from extant species like Lachauncia berry or Zygosporilospora maracii will actually complement my mutants and test whether this is the case, that these particular lineages have nanoproteins that have distinct functions. However, the thing that I think I'm most interested in trying to test is whether or not there's been some kind of subfunctionalization that's happened. Essentially that these cell wall nanoproteins may have multiple functions. We're not sure what those functions might be. They could be something like facilitating sterile transport, as well as a separate function of contributing to cell wall integrity or something. And the idea here is that after the whole genome duplication event happened, you have multiple genes that are now sort of relieved from selection on both of their roles. And so they sort of specialize into perhaps tier 1 having one of these functions, and then tier 3 ultimately arriving at a different function. But they sort of do them better than the ancestral protein had done both of those functions before. And so I'm thinking of testing this out as well as a model of neofunctionalization, where perhaps a very similar thing has happened, where after the whole genome duplication event, these strains now have multiple cell wall nanoproteins that are able to sort of evolve a whole new function. And so potentially tier 3, which we know to be this essential gene in Saccharomyces cerevisiae for anaerobic growth, perhaps this is something that sort of evolved later because it was able to sort of freely evolve now that these nanoproteins have been duplicated. And so these will be fairly difficult to test, but we're interested in trying these out, determining whether essentially some of these ancestral or diverged lineages have particular tier 1 or tier 3 functions. And I should actually say, as I'm sort of closing up here, that when I talk about this functional divergence, we know very little about what this actual function is. There's very little done on these cell wall nanoproteins to know what they're actually doing. We just know that they're the outermost layer of the cell, that they're heavily manosylated, but that is about it. And so I'm really eager to use budding yeast as a really diverse system from which we can look at a lot of these different alleles of these genes, and then perform these tests in Saccharomyces or other species, because there's a lot of species of yeast that are really good heterologous systems for expressing these different genes. And so I'll close on this. I had mentioned it a little bit before, that there's this distinct lineage of budding yeast, the Brettanomyces, that are capable of growing anaerobically. And what's fairly interesting, and I don't really have time to talk much about it today, except for here, is that there's a very similar looking pattern in the Brettanomyces, in that its obligate aerobic relatives also have these cell wall nanoprotein sterile transporter gene clusters in their genomes. And in the Brettanomyces, the lineage that is capable of growing anaerobically, we see a lineage-specific amplification event of these cell wall nanoprotein genes. So they're not necessarily linked to any whole genome duplications, but we do see that while most of these species related to the Britannomyces have 1 or 2 cell wall nanoprotein genes, the Britannomyces species that we've sequenced have 4 to 6. So something might be going on, we think, that might be parallel to what happened in the Saccharomyces case, that these cell wall nanoprotein genes are undergoing amplifications in their copy number, and somehow that's enabling sterol transport either to evolve under a different regulation or just to evolve to become better in these lineages. And so this is sort of my final current model for how anaerobic growth evolved in budding yeast. My idea right now is sort of you have an ancestor that has these genes for sterol transport, whether that's the transporter itself, PDR or AUS, and a cell wall nanoprotein. And maybe it's inefficient, but it provides benefits when sterols are prevalent and oxygen becomes scarce, although not when it's completely anaerobic, and sort of takes the pressure off of needing to get that oxygen to make these sterols. And ultimately, somehow these cell wall nanoprotein loci are amplified, creates this increased gene dosage at first, but ultimately offers some opportunities for these to functionally diverge. And maybe that enables sterol transport to increase to levels that actually then can replace biosynthesis altogether, at least under particular conditions. And then ultimately, there's some kind of regulatory rewiring to induce these under anaerobic conditions and sort of downregulate their aerobic counterparts. And that's sort of my way of thinking right now about how anaerobic growth evolved in budding yeast, at least the sterol transport side of it, I should say, since it's still likely a fairly complicated phenotype that has arisen in budding yeasts. And so with that, I'd like to thank everyone and take any questions. Yes. Okay, so the question was whether I think that the difference in the function of tier 1 and tier 3 are both regarding sterol transport, or whether they may have maybe functions not related to sterol transport. My current thinking about it is that I don't have a lot of great evidence. It's sort of just a gut feeling at this point, but that perhaps tier 3 might be involved in sterol transport, for example, but tier 1 and some of these other cell wall nanoproteins may be involved in some structural role for the cell wall, or that's my best bet. It's really hard to think about what functions these cell wall nanoproteins have, and so a lot of the thought is that they're involved somehow in the structure of the outer part of the cell wall. And so perhaps I could envision a scenario where one of these nanoproteins evolved to facilitate a specific transport mechanism, and then the other one sort of evolved to take on more of a structural role without needing to facilitate that other process. So the question is whether there's anything that you can glean from the sequence comparison of the amino acid level. And the problem is that the major difference in their amino acid sequence is those really long stretches of serines and threonines. And so that seems to be where the main differences is between them, but it's really not clear what that does. Yeah. Yes, they're very tier 1 and tier 2 are very similar to one another. Yeah. In terms of the length, for sure, I would have to go back and see whether they have the exact same motifs and things going on in that region. Yeah, so if you overexpress tier 2, it can compensate for tier 1. And I would suggest that might be because if you look at some of the RNA-seq data that's out there publicly, it's because tier 2 is a lot at a lower expression level than tier 1 is. So that might explain it. I think that they have a common function just based on their high relatedness, and yeah, I think tier 1 and tier 2 probably have overlapping functions because they are so similar to one another and have similar genetic effects. Yeah, thank you. All right, I have a couple questions from the chat here. Oh no, I went. Yeah, so thanks, Chris, for the question. How might I design an experiment to test the function of the tier manoproteins? So this one is definitely a complicated question because I'm really interested in the function of these things, but I am not really biochemically oriented whatsoever. But if I could take a stab to just say what I'm thinking, I do have interest in doing some experiments to parse out what parts of these manoproteins are important for their particular functions. So I mentioned, if I can kind of see if I can jump back way back to the sequence and just the simple sequence analysis. I am curious whether we can at least initially parse out whether this N-terminal region that is sort of has actual complexity to the sequence and is fairly conserved is distinct in the function that it provides to this sort of low complexity C-terminal region that has all these serines and threonines. And so one of the first things that I think I might try to do to assess functionality is figure out whether different parts of the proteins have different functionalities. So essentially, if I were to link up the N-terminus of tier 3 with the C-terminus of tier 1, for example, would I get some kind of cool hybrid chimeric protein that does both functions? Or perhaps if I get rid of so instead of deleting tier 1, for example, what if I just turn tier 1 into something that looks like the C terminus of tier 3 or the N terminus of tier 3? And would I abolish anaerobic growth in doing that? And I think we might be able to start to get at where the function actually lies in the protein. But ultimately getting to the function of the proteins, yeah, we'd have to apply some clever biochemistry. My mind is always more genetically focused, and so I can think of ways we might perturb this system to see what aspects of these proteins contribute to anaerobic growth. The exact mechanism, I think, is really interesting, and I'm going to have to chew on it quite a bit. But thanks for the question.
I'll ask a technical question. I just thought of Euro 1. The need for Euro 1 would only be strong if uracil was not present in the medium. Was your anaerobic growth there in minimal medium, or was it in switches that you're doing that have uracil?
Right. So Chris asks whether whether my anaerobic growth medium has uracil in it, in which case, at least for the anaerobic growth experiments, I'm sort of relieving the selective pressure on uracil. And so basically, why do the Nicasiomyces grow anaerobically without Euro 1? And yes, I have uracil in those media. I've tried to grow them without uracil as well to mixed results, but I need to replicate those experiments and sort of do them more consistently. This was several years ago, data that had some other issues, and so yeah, they otherwise have a when I say that they've lost Euro 1, they do have the aerobic version, the Euro 9, this aerobic analog of uracil biosynthesis. So they aren't complete uracil auxotrophs, but they should, in theory, become one under anaerobic conditions. Yeah, thank you, everyone.